My name is Michael Kudinoff, and I'm an assistant professor of electrical and computer engineering at North Carolina State University. My research is primarily focused in sensor development and systems development, so combining multiple optical elements together and electrical circuits as well to create new kinds of optical sensors. I guess my interest in science and engineering primarily came from my father, who was also a scientist. The applications that my research focuses on presently involves biomedical imaging, so for instance disease detection, sending light into tissue and getting the reflected light, measuring the reflected light in some way to investigate changes in that reflected light due to things like cancer or the presence of inflammation, for instance. Another application we're looking at is in farming. So imagine being able to take these small, very lightweight, unmanned aerial vehicles, equipping it with this very miniaturized spectral imaging camera, and being able to identify which plants have disease, what kinds of diseases those plants have, what kinds of plants they are. Light is physically composed of different wavelengths. And so what is a wavelength? A wavelength is a different color of light. So if you ever spread light out using a prism, so you see different colors, blue, red, green, spread out onto the wall, those correspond to different frequencies of light. It's an oscillating wave traveling at a certain velocity, speed of light, and that oscillation occurs at different frequencies for different colors. And specifically, blue light has a higher oscillation frequency than green light. Green light has a higher oscillatory frequency than red light. So red light, to our eyes, is really the lowest energy or longest wavelength that we can see visually. And by studying the properties of light interacting with matter, specifically by measuring how light reflects off of different objects, we can quantify different aspects of properties of tissue, for instance. We can use it to diagnose disease. We can also use it to identify the health of a plant. Perhaps is infected with insects, or perhaps it's being attacked by a fungus. And so all of this information is sort of encoded inside this reflected spectrum. And what we're after is what specific signatures can we use, what specific wavelengths of light can we use to identify those diseases or those funguses. So to quantify something is to measure something, basically trying to establish a relationship between what you're observing and what's actually happening. So we have these wavelengths from blue to red that we can visually measure with our eyes. However, there are other wavelengths of light beyond those that we can see. For instance, wavelengths that are shorter than blue light, in other words, we have a higher oscillating frequency, shorter than blue, correspond to higher energies like X-rays or gamma rays. So if you go to the dentist, for instance, they're basically shining these really high energy, very bluer than blue, rays of light into your body. And they're doing that for the purpose of being able to image your teeth. On the other end of the spectrum, towards the red, if we go to even longer wavelengths than those that we can see, we call that infrared. And so all these other wavelengths of light contain additional information about the target. And so part of the research that we're doing here at North Carolina State is also looking at how these longer wavelengths can be used in these small imaging cameras that we're building for agricultural applications. So just as you have your own signature or your own unique way of signing your name that ties your signature to you, we can also find unique signatures in light that ties the different observations that we're trying to make to a particular spectrum. An example of that would be, say, we're trying to identify a specific chemical, like water. We can shine different wavelengths of light at the water, measure the light that we're seeing in, say, reflection or transmission. And that light is essentially imprinted with a specific signature that allows us to identify, yes, this is water. This is my lab. This is where I and my students do most of our research. And specifically what we're interested in, again, is characterizing different chemicals or objects or phenomena. This system here we refer to as a spectrum generator. And it consists of this light source here, which is a gas discharge lamp that contains xenon and mercury in it. And that light is focused onto a small aperture here. That focus light is then collimated into a diffraction grating. And this little grating here actually acts just like a prism, and it spreads the light out into its colors. And actually you can see the colors on this electronic chip here. And what we're able to do is selectively reflect light from this location into this, what's called an integrating sphere. So this sphere essentially takes that light that we're selecting here and homogenizes it, smooths it out spatially, makes it more spatially uniform. And this is the light that we're most interested in observing with our instruments or our sensors. So we can use this light to calibrate our instruments, which enables us to make our observations in the field. And actually over here is another kind of spectrometer. In this case, this spectrometer just takes light from the sphere here and guides it through an optical fiber into another type of diffraction grating or prism type element. And so that light gets dispersed into its colors, and then we measure it on a camera. Essentially there's a small camera in there. And the output of that camera is displayed on this computer. And here you can see the different colors of light represented as numbers from 350 to 800 nanometers. It's essentially the distance or the wavelength of the light. So this is a view of the spectrum here. So this is essentially power versus color or wavelength or frequency. And then in this particular spectrum, there's more blue light and red light than there is green light. There's almost no green light in this particular spectrum. And so if we go over here to the control software that allows us to load different spectra, we can actually browse from a library of different spectra that we can load into the chip. And so if we pick one of these spectra that has just this bar going down through the middle of them, we get a single wavelength of light. So in this case, if I open this file and load it into the chip, it'll change the spectrum here to only blue light. So we only have blue light present in the sphere. And so this effectively is being done, in this case electronically. In your case, it will be done with the spectral filters that you'll be designing. This instrument here allows us to measure these longer, lower energy wavelengths. And it's called a Fourier transform spectrometer. It essentially measures the spectrum in a way that's slightly different than the system that you just saw. The system that you just saw spread the light out into different wavelengths or different colors. And those colors were mapped to different spatial locations on that chip, and that's what allowed us to pick different wavelengths. In this instrument, we're actually creating interference between two beams of light. So we send in one beam, that beam gets split into two beams, and then we recombine them and create interference. And it's the interference that allows us to measure what wavelengths of light are present in the source. Longer wavelengths of light actually correspond to heat energy. So you're actually emitting light right now in the form of heat. And in a detector, when we're actually trying to measure light, that's a bad thing. We don't want to be able to measure the heat coming from the detector. And so in that case, we actually have to cool the detector down using liquid nitrogen. And so this is the liquid nitrogen, and we can pour it in. And so once that's cooled down, we're actually able to now measure these much lower energy photons, much lower frequency light. So now we're going to have you create your own spectral database by creating an array of different spectral filters, of your own spectral filters. And this is an example of 6 different filters that you may use to create a spectral database. And then you'll be given an unknown filter, and you'll have to match that filter to one of these filters that you have in your database.